Um, at this point, you may also be thinking, I've been talking a lot about the historical people that have worked on rust fungi. I haven't really talked about my credentials with rust fungi. And honestly, I don't have that many credentials with rust fungi. I'm the Purdue University curator of fungi. That's about it. My background is not in rust fungi. But the reason I believe Kathy hired me was because I have experience with culture collections, and in part of working in this role, of course, I'm working with those 7,000 cultures in the AIM Lab Culture Collection. But my background is working on several different types of fungi that have been culturable. Um, so I may be a curator now, but in the past, I've just been a systematist or a taxonomist. While I was at the University of Maine during a postdoc, I worked on uh, a new Ophiocordyceps species that was infecting invasive European red fire ants. Um, while I was at the University of Florida, I worked on Basidiomycete and Ascomycete fungi that were in association with ambrosia beetles, both in uh, Florida and Taiwan. But during my graduate programs and other postdoc programs, I've been working fairly extensively and exclusively on zoosporic eufungi, also colloquially known as chytrid fungi, and I'll get into all of that. But specifically, most recently, I was at the University of Michigan, where we established the Collection of Zoosporic Eufungi at the University of Michigan, or the CZEUM. And this is a cryopreserved collection of these culturable zoosporic eufungi that have been accumulated thanks to the donations primarily by Dr. Joyce Longcourt at the University of Maine, with whom I received my master's and PhD, and also Martha Powell at the University formerly at the University of Alabama. So I'm already talking about zoosporic eufungi. What are zoosporic eufungi? Zoosporic and makes you think they can have tails and swimming, and eufungi, true fungi that can swim. Let me tell you a story. This is my son, Jasper. In this photograph, he is 7 months old. This was about a year and a half ago. And I determined that at 7 months, it was well beyond the time that he should know what fungi are. So we got out his first book of microbes, and you can see there the third subject in this book was fungi. So we're in for a good story. Um, However, when I turned to the page of information on fungi, the second sentence was false. And that second sentence is fungi have no cilia, small hairs, or flagella and cannot move around by themselves. And that is the basis of my entire PhD program. Um, so I was rather upset. And the thing that really got me the most was they used all the right terms. So they didn't Google anything. Because my first course of action as a sensible parent was, of course, to go on Twitter and to contact the authors and tell them about some recent publications, which they probably would have found if they had done some Google searches, that had emphasized that a lot of these early diverging lineages of fungi or non-dikaryotic fungi, they do have flagellated life stages. You may see in that tweet that it received 17 views, not 17 likes, 17 views. So I'm always hoping to expand my audience a little bit more when I talk about zoosporic eufungi. So again, what are zoosporic eufungi? As I said before, they are sometimes called chytrid fungi or chytrids. I know I slip into that a lot, but chytrids are actually just a subset of what we now understand to be zoosporic eufungi, but historically, that was what we called all of them. So if you may find me slipping into that as well, still. Um, they are generally saprobic, but there are very notable exceptions to that. But the defining feature really is that zoosporic stage. It's bound by a plasma membrane that lacks ergosterol. There's typically a single smooth posterior flagellum, but sometimes they can be multiflagellated in certain phyla. And these are produced in some sort of reproductive center called the zoosporangium or multiple zoosporangia over the course of a single thallus. And these things have been around for a while. The first one was described in 1851, Chytridium ola. Um, there have been very prominent researchers and presidents of the Mycological Society of America that have worked to bring these things into culture for a better understanding by researchers and just general biology courses and classes. And there's still people that are working on getting now the anaerobic ones into culture so that we can further appreciate and understand them and really study them to the fullest degree. But perhaps the two of the most prominent names within the field historically have been Fred Sparrow and Alma Whiffen Barksdale. Sparrow literally wrote the book on these organisms. He was at the University of Michigan primarily, and his book is massive, probably 4 or 5 inches thick. At least the 1960 edition, 1943 is a little bit smaller than that. And in his book, he divided these organisms, which were at that time just in the Chytridiales, that one order. He split them based on whether or not they were operculate or inoperculate. If they had this cap-like structure at the apex in general, that would pop off and release the zoospores from the zoosporangiums to start the entire asexual reproduction process over again. Whiffen did not believe that was the best phylogenetically informative system, and in her 1944 rebuttal, she proposed that the type of development was more important. And by the type of development, she was referring to endogenous development and exogenous development. In endogenous development, the zoospore will find a substrate that it likes, it will insist, and it will producing a cell wall of chitin, and the nuclear material will stay within that cyst, and the zoospore cyst will essentially just continue to swell to form a new zoosporangium. So you have the nuclear material never leaving, it stays within in endogenous growth. In exogenous growth, the nuclear material goes outside of that zoospore cyst through some sort of germ tube or hypha-like structure and forms a zoosporangium elsewhere on the thallus of the fungus. So she believed that this was a much more phylogenetically informative character upon which to start to split these organisms. One proponent of Whiffen's work was John S. Carling, and this is where I tie it back to the University of or Purdue University Herbaria a little bit instead of going off on this tangent. He was at the Purdue University Department of Biological Sciences from 1948 to 1958. He came here from Columbia University, where he originally worked on algae and became interested in chytrids when they kept parasitizing all the algae that he was trying to study. But he agreed with Whiffen that endogenous versus exogenous was a much better system than operculation, as Sparrow had recognized. Carling is a very interesting person. Again, if you go on the Purdue University Herbaria website, I have written an entire biography about Carling. Just the short summary is that he is a White Sox reject. He accidentally rediscovered some Myositis while he was lost in Mexico. He had a rivalry with President Dwight D. Eisenhower that started at Columbia University when he was president there. So he's just a very interesting person. Please check out that biography. But pertinent to today's talk, he wrote this book in 1977, Chytridiomycetarum Iconographia, and it is kind of the companion piece to Sparrow's work. Sparrow was very wordy, extensive, perhaps even exhaustive, but Carling liked to draw, and every other page of this book is illustrations, beautiful illustrations of these organisms, kind of bringing them to life for people that don't have access to microscopes or even just an awareness of these organisms. So Sparrow, in his second edition of Aquatic Phycomycetes, tried to tried to talk to Whiffen a little bit and say, yes, I understand this is a somewhat artificial system, but your system still doesn't work. There's too many types of variation development, whereas it's always a dichotomy of operculation versus inoperculation. That's just how it is. Carling in 1977, though, said that's not true. There's 30 years of evidence that show that that's not true. And this is just a largely arbitrary system that you find works best for you. Let's just agree not to agree. and they pretty much didn't. But between 19 starting in the 1960s, there were other forms of information that were being generated to help in aiding the taxonomic systems of these organisms. So much so that by the late 90s, we had 5 orders of zoosporic eufungi. The Chytridiales we have already addressed a little bit. The Blastocladiales and the Monoblepharidales also previously existed based on primarily on sexual behavior that was very easily distinguished from the Chytridiales. The Spizella mycetales was the first order that was described based on electron microscopy ultrastructure of the zoospores. So we had this new line of evidence that could be used for delineating taxa. And the Neocalamastagales were these polyflagellated chytrids that were growing in an anaerobic guts of ruminants. So they were kind of in a complete separate niche and very easily recognized as a separate lineage. So in the late 90s, though, there was this complete explosion of interest in chytrid fungi and in zoosporic eufungi, thanks to the description of Batrachochytrium dendrobatus, or BD, the first amphibian pathogen, for the first vertebrate pathogen of chytrid fungi. And this discovery by people at the National Zoo in Washington, D.C., and also people in looking at wild populations in Australia, was facilitated by Dr. Joyce Loncourt, who was the first person to get BD into pure culture at the University of Maine. And it really led to people actually knowing what a chytrid was. If you've heard of a chytrid, you've probably heard of this chytrid, or you've heard it referred to as chytrid, and just assume that there's just one chytrid in the world, but there's actually 1,000 or more. So much was this brought to the public's attention that there were children's books written about it. The Case of the Vanishing Golden Frogs. In the middle is some album art for a recent album just came out this past year called Chytridiomycosis Relinquish. That's by the British death metal band Slime Lord. So it's reached that corner of the world. There's the Chytrid down there in the bottom corner. That is actually a video game that you can play as a frog determining a murder mystery. So it's pretty well ingrained in people's minds at this point that they have a probably big misconception on what a chytrid fungus is, but they're at least aware of the term. But in the middle here, this map of Costa Rica and Panama, I want to emphasize this because this was the first instance in 2022 of it being shown that this tiny little microscopic fungus that's single-celled zoosporic thing, it has human health implications. This is a map showing the expansion of Chytridiomycosis, the disease caused by BD, which causes amphibian die-off in over 500 species, or at least affects 500 different species of amphibians. This shows Chytridiomycosis sweeping across those countries. And in this paper, they correlated that decline of amphibians with increases in malaria cases. Because as you have the frogs dying off, then you have the mosquitoes going up, and you have the malaria going up in humans. So again, this little microscopic fungus that we didn't know anything about until 1999, it's a pretty big player in the world if you look in the right places. And none of this work could have been accomplished without the great isolation efforts and just many decades of work from Dr. Joyce Loncourt at the University of Maine and Martha Powell at the University of Alabama. So with these massive culture libraries generated by others, especially Dr. Powell and Dr. Loncourt, people like Tim James at Duke University were able to make these phylogenies to really expand upon our knowledge of what it is that we need to do now with the Chytridiomycota, or the chytrid fungi, or the zoosporic eufungi. In this left phylogeny, this was the first phylogeny published in 2000 that showed that the Chytridiales was polyphyletic. There are multiple different lineages within that order. And the center phylogeny shows that these different clades could be correlated to ultrastructural evidence within the zoospores. So we had those lines of evidence that could support the description of new taxa. Finally, on the right side is a phylogeny of the entire fungal kingdom that shows that this zoosporic eufungal state of flagellation, it didn't evolve once, or if it did evolve once, it's been lost multiple times. There are lineages that are not just in this one monophyletic clade that still possess flagellation. So there was a lot more biodiversity to be had, even at the degree of phyla. Dr. Pete Letcher at the University of Alabama, a student of Martha Powell, he equated this to waking a sleeping giant, that we had all this work to do, thanks to specifically to the isolation efforts of Dr. Longcourt and Dr. Powell, but also to the molecular work of Tim James. So we started naming new orders left and right. Eventually in 2000, from 2006 to 2014, we named 8 new orders. And these are based primarily on those collections of zoosporic eufungi that we still have at the CZEUM in Michigan. While I was a postdoc in Michigan, while we were forming the CZEUM, we also were checking these cultures for viability, thawing them, making sure they were alive, and then putting them back into cryopreservation. And while we were doing that, all the ones in blue in this phylogenies are ones that we got to grow, or that we had proven viability within the CZEUM. And while we had them out and growing, we went ahead and did DNA extractions and produced this comprehensive phylogeny of the Chytridiomycota, the Monoblepharidomycota, and the Blasticladiomycota, to show where we still have questions, what lineages still need work. And we did this through an examination of ribosomal DNA long reads generated from Oxford Nanopore Technologies. We ended up getting over 400 sequences from over 400 strains to produce this phylogeny. So all of the ones in red, however, are cultures that have been unfortunately lost over time. But this is all still just the cultured material. There's so much more that is still being found when people are looking at environmental sampling, and they're finding lineages that just don't map onto anything else that we have. But luckily, there are people like Kensuke Seto in the world that are working on those organisms. Kensuke is just a master at looking at a single algal cell, determining if it has a fungal infection, and doing mouth pipetting to pick it up, doing single cell sequencing of that specimen in order to get a molecular sequence, and in some cases actually getting it growing in dual cultures so he can do electron microscopy of those zoospores as well. You can see in this work, he's actually described a couple of orders based on his analyses. And what's particularly impactful from Kensuke's work is he went from photograph to sequence. So he knows what that sequence was doing in the environment when he found it. And a lot of these environmental sampling assays, there's really just conjecture about what they're actually doing in the environment. We know they're there, but are they even biologically active, or was that just dead material that washed from upstream? We have no idea. But Kensuke knows exactly what sort of alga or other organism this thing was parasitizing before he put it into the phylogeny. And I think as time goes on and we find more and more environmental samples, we'll be able to properly understand what it is that they're most likely doing in an environment, thanks to people like Kensuke. We also have orders that are being described that don't follow the rules of zoosporic eufungi, which already aren't following rules. This is a new lineage of Chytridiomycota, new order, and it's lost flagellation. So that's not unheard of. We already knew of some zoosporic eufungal lineages that didn't have flagellation, but to have this one specifically be like in such a strong relationship with in the parasitizing insects, it's just so odd that we're still finding these things again, that zoosporic eufungi that are already breaking the rules of mycology and fungi that are now these sublineages that are breaking the rules of zoosporic eufungi. So I think as we look more and more, we're going to find more instances like this. Finally, with the cesium, one of our biggest goals while I was there was to understand how phylogenomics of these organisms could be utilized to understand and appreciate how they're properly related. Based on the ribosomal information that we had, we didn't really know the full backbone of a lot of these phylogenies, which orders were related to other orders, how the phyla were really shuffled around exactly. So we set out with the goal of examining some choice cesium cultures to determine this relationship based on phylogenomics. But it really didn't answer the question. It gave us pretty much the same answer that we'd been getting all along, and there was just so little support for any strong hypothesis. However, it did kind of answer a question that we didn't even think about asking when we started, which was, are these things haploid or are they diploid? It's just widely been assumed that haploid zoosporic fungi just give rise through the evolutionary chain to higher diploid dikaryotic fungi, the Ascomyces and Basidiomyces. And these quote unquote simple fungi back here with the zoosporics, they of course were just going to be haploid. They reproduce asexually. That's just how things are. But with this analysis of the genomes, we actually showed that it's more common for these organisms to be diploids and to be asexually reproducing diploids, with rare instances of haploid behavior and perhaps sexual recombination that, for the most part, we don't see take place, at least in pure culture. So it kind of upturned that assumption of that things go from simple to more complex. It actually turns out that things were already pretty complex to start with, and they may have actually gotten a little bit more simpler as you went up the evolutionary tree. I'm sorry, and this is Tim James with the lead author, or Kevin and I were co-lead authors on this project, but this is Kevin Ames is doing his best Tim James impersonation. So why did I just hijack 20 minutes of this presentation to talk about chytrid fungi or zoosporic eufungi? And I think that really the answer is we have to emphasize the importance of collections in all forms. I took a very loose connection to John Carling and turned it all about chytrid fungi. But really, for me, the cesium at Michigan, the Purdue University herbaria here, they're all in the same boat. They're all showing us that there is so much information to be gained from these organisms. We can't lose these organisms. We need to properly examine them, continue examining them through new methods, determining the true biodiversity that we have within these systems. And if we just start to dispose of these collections, that's just not going to help us in the long run. In the case of the zoosporic eufungi, we've gone from Carling and Sparrow and Whiffen arguing about a single order of the Chytridiales to then our understanding of electron microscopy leading to the description of 5 orders, to then understanding that through molecular evidence that there are correlations to be made with all of this previous information. But now on the right, we recognize that within the Chytridiomycota, there's probably about 15 orders, and exterior to that, there's another 6 phyla that possess zoosporic eufungal lineages. So really, it just really shows the emphasis that none of this could have been possible without collections in some form or fashion. So I just wanted to hopefully bring that to people's attention today. But with that, I will return you back to Herbaria and say if you have any other information or questions that you'd like to obtain or answers for, you can contact me. You can visit the Purdue University Herbaria. I am also the webmaster of that, and if you have any suggestions or things you'd like to see, please let me know. And again, I emphasize that Purdue alumnus are article. It's just visually stunning. They really did a great job making us look good and the herbaria look interesting to people that maybe aren't that interested in the topic to begin with. So hopefully we'll get some more people in the door. And I thank you for your attention.